We'll now be discussing a topic I'm sure you've all been anxious to talk about, and that is respiration. We'll move on to photosynthesis after that. Respiration, of course, is the way that living organisms make ATP from sugars like glucose, and glucose is the main one we'll be talking about. On this slide, you can see a light micrograph of a mammalian fibroblast that's fixed and stained with fluorescent antibodies. The mitochondria are denoted in green and microtubules in red, and you should notice the concentration of mitochondria, especially around the nucleus, and the green showing through there is mitochondria that are under the nucleus, and this is one of the most metabolically active parts of the cell, and because it's metabolically active, it needs ATP, so notice where the mitochondria are concentrated around that metabolically active part of the cell. Here you see mitochondria in a living cell, a living fibroblast, viewed with phase contrast microscopy, and you can see that they're much longer than you usually think about when you see the little sausage-shaped mitochondria, and that's because they can fuse together and then separate as well. They are much more dynamic than we've been led to believe, as I mentioned just a few minutes ago. This is an electron micrograph of a mitochondrion, typical of the ones you see frequently. Notice the cristae, which extend across the mitochondrion. Also notice the double membrane on the outside and the sort of grainy appearance that you see inside not inside the cristae, but sort of outside the cristae, but inside the double membrane. A lot of that grainy appearance is actually ribosomes that are responsible for making the proteins that mitochondria need. Here you see the mitochondria of a sperm cell, and quite often these mitochondria fuse into one giant mitochondrion that spirals down the neck of the sperm cell right below the head of the sperm. And you can see that here. You can see it sort of coming into and out of the plane of focus of this slide. And again, this is an electron micrograph. In this schematic drawing of a mitochondrion, take note of the inner and outer mitochondrial membrane and the space in between. That is one of the compartments in a mitochondrion. The other compartment inside the mitochondrion is the mitochondrial matrix, where you find ribosomes and sort of the cytoplasmic-like interior of the mitochondrion. And also you'll find the mitochondrian's circular DNA inside that matrix. Also take note of the cristae that extend into and across the mitochondria. They provide the folding of those cristae provide more surface area for the production of ATP, the enzyme that makes ATP called ATP synthase, and that used to be known as lollipop structures, extend from the cristae into the mitochondrial matrix. As I alluded to a few minutes before, mitochondria can undergo fission, that is, they can split apart, and here you see some mitochondria involved in that process, splitting in half, much like bacterial cells do. And remember, mitochondria are derived from bacteria. That is what their origin was, according to the endosymbiotic theory, which is now simply known as endosymbiosis. This is another view of a mitochondrion. This is a scanning electron micrograph of a macerated mitochondrion. And what's nice about it is you can sort of see the three-dimensional look of the structure. And here you see a 3D reconstruction of a mitochondrion based on high-voltage transmission electron micrograph pictures that have been taken of a mitochondrion. And you again get a more complete view of what the mitochondrion looks like. This schematic drawing again emphasizes the two compartments inside the mitochondrion, the space between the two membranes, the inner and outer membrane, and also the space inside the inner membrane, which is the mitochondrial matrix. You also see in this drawing the ribosomes, which are the little sort of blue-black dots that you see spread throughout, and again, the orange things are the what were originally called the lollipop structures. When they were seen in the electron microscope, they looked like lollipops. I'll show you a picture of one of those later. But then eventually it was discovered that these were actually the molecules that were responsible for making ATP, and they're now known as ATP synthases. Here you see the overall scheme of respiration, the three parts: glycolysis, Krebs cycle, and electron transport. Glycolysis occurs in the cytoplasm of the cell, and in the presence of oxygen, the products of glycolysis go into the mitochondrion and are further broken down. In the absence of oxygen, glycolysis passes its products on to fermentation to extract some of the energy from those molecules. Obviously, in the presence of oxygen, you can extract more energy. We'll talk about each of these processes in sequence, starting with glycolysis.